right, I think I started that going. All right, thank you, everybody. I am painfully aware that I'm the one thing standing between you and lunch, and that this is a really long morning to spend sitting. So I'm going to suggest, no, in fact, I'm going to insist that instead of you all sitting politely and saving your questions for the end, that you interrupt me along the way, and we make this a little bit more interactive. I think that'll keep us all awake, and make the time go. First, just to get a sense of the audience, and you're allowed to raise your hand more than once. How many people here are working on mammals? Let's got a lot of mammal people. Birds? A couple birdie types. Amphibians? One amphibian. Reptiles? Okay. Fish? Got some fishy people. Plants? Very good. Invertebrates? Very good. Okay. Other? What's other?
Microbes.
Microbes? Okay, very good.
Microbes.
Microbes. Okay. I forgot the microbes. Sorry about that. Okay. Did I miss everyone? We got everyone's taxonomic group. Okay, that gives me a sense of who we are. So the work that I do. Okay, here. Oh, you know what? Did we take the USB chip out of your computer and put it into mine?
No.
That's why it's not.
It's not in yours.
Otherwise, I don't think this is going to work. You got to keep an eye on these people. Okay, let's see if this works. Okay, that's better. Okay, so the type of work that I do is in ecotoxicology and ecology, primarily aquatic, and I say primarily because now a lot of our work involves modeling, and one of the advantages of doing modeling as opposed to wet lab work, it does open opportunities to work on lots of different taxonomic groups. So in my group right now, we're doing some fish stuff, we're doing some plant stuff, we're doing some invertebrate stuff. I even had a visiting scientist a couple years ago who was doing some bumblebee stuff with Nika and I. So you can do a lot of different things. The reason that we do this kind of work is not just to improve our understanding of some of these processes, but we actually want to help inform decision making, and in particular, inform how we assess the risks of toxic chemicals and how that can inform management decisions. And a lot of what I'm going to talk about, and a lot of our research, is focused on chemical stressors, but most of what I'm going to say could apply to any kind of stressor, whether due to climate change, as Lauren pointed out. Climate change effects can appear in many, many different ways, and a lot of the models that we use could really apply to any type of stressor. So I hope you'll keep that in the back of your mind, even if my examples are somewhat chemical. And we're doing a lot of mechanistic modeling to help both understand and predict the effects of stressors on organisms. And I should say, historically, ecotoxicology has focused pretty strictly on chemical stressors, but I think it's becoming more widely recognized that chemicals don't exist in isolation, either as single chemicals. Most times we have complex cocktails of chemicals to which organisms are exposed, but also they exist in combination with other stressors, not the least from climate change. So the work involves some combination of estimating exposure to our stressor, in this case maybe a pesticide, emphasis on toxicology and toxicity, largely because the field evolved from human toxicology. And increasingly, and what we're trying to do, is bring ecology into this picture and look at how all those things that aren't just the toxic response of an organism to a chemical, but how the environment and interactions in the field influence the responses of organisms to toxic chemicals. Whereas Jonathan's talk was mostly focused on genomes to phenomes, mine is going to be more phenomes to populations. But I think there are a lot of parallels between the challenges that we face in doing this kind of work to what Jonathan talked about. In our field, in ecotox, what we typically measure, we do toxicity tests, and historically, most of the data that we collect is on mortality. You know, you put your critters in a beaker and you expose them to a chemical and count how many are dead at the end of the experiment. And that's historically what we've measured, but that's not really what we care about. What we care about is the persistence of fish populations and increasingly what those populations do for us. And we've begun to articulate these protection goals, if you will, in terms of ecosystem services. So all the things that organisms do in nature from which humans benefit. And these might be, you know, enjoying some recreation or aesthetics, in this case, you know, recreational fishing or boating. There are lots of different ecosystem services, many of which, if not all, are performed by populations of organisms. So I think the link to populations is really important here. Increasingly, because of the technological advances, we're measuring things at lower levels of organization. So instead of measuring individual performance, we're measuring all of the omics, right? So the genomics, and now we can do transcriptomics, and proteomics, and metabolomics. We can generate a lot of data at these lower levels of organization. The problem is that these responses to exposure to chemicals are even further removed from what it is we care about and what we want to protect than the individual organism performance. And so how are we going to link what's happening at the suborganismal level to what's happening to whole populations of organisms is one of the major challenges we have, and we need models to do this. And that's the kind of modeling that we're going to talk about today. What we've done in the past is kind of use proxies. So we've measured toxicity, and we look at the responses of individual organisms to stressors, and we've said, okay, you get 50% dead or 50% reduction in growth rate or some physiological process. And so implicitly we've assumed that, well, that's kind of a proxy for what's going to happen at the higher levels of organization. So you might do, come on now, work with me. There we go. We might measure toxicity, so you'll have some kind of toxin or stressor concentration on the x-axis. You do your test and you measure, in this case bees, how many bees die. And it'll be some nonlinear shape, usually some kind of S-type dose-response function. That's the data we typically have. But we can't just assume that if you get 50% dead bees, you're going to have a 50% impact on the population of bees, which is what we care about. Typically, I see what you mean, Jonathan. This thing is really sensitive. Yeah, I need longer arms. Because typically there'll be some relationship between your mortality of individuals and, say, population size or other measure population dynamics, and it's not going to be linear. In some populations, you could get very large decreases in survival before you start to see a decline in population size because of internal mechanisms going on, compensation in the population. Others, you might see quite a rapid decline in population size with small increases in mortality. So we can't assume that your individual responses are directly proportional to your population level responses. And likewise, if we want to go to an even higher level, let's say we're not interested in the size of bee populations per se, but we really care about pollination. And we want to make sure that if we do agriculture, the effects on the environment don't reduce pollination. Well, then we kind of have to figure out, well, then how does the size of our bee colonies or other properties of those populations, how does that relate to the process of pollination? And again, those relationships aren't going to be linear, so we can't assume direct proportionality, so we need some kind of models to make the linkages from one level to the next. Here's one example of a model that we developed of a threatened plant species, the decurrent false aster or Boltonia decurrens, and we were looking at effects of flooding, competition and herbicides on its population dynamics. And so here's a good example where we have chemical stressors in combination with various natural stressors. So we developed a population model. This is an example of an individual-based or agent-based model where we have different life stages, so seedlings, rosettes, which is a vegetative stage, flowering plants, and then seeds that can stay in the soil for years, and then different processes. So the seedlings grow, you have rosettes growth, the plants mature at some stage, reproduce vegetatively, and then they make seeds. So these are both the stages and the processes that we were able to incorporate in the model. And the area surrounded in blue are those processes that are affected by various environmental factors. Now, in this particular species, and here are some results, this particular species needs flooding to survive. And so here you can see some of the results. The black lines are the population size. This is in years, up to 15 years, number of plants per square meter on the y-axis. If you've got flooding in the black, the populations are pretty stable. If you have flooding every winter, they love it, they're happy. If you don't have any flooding, regardless of whether or not you have herbicides in the picture, and the dotted curves are with herbicides, the solid ones are without, your population here in the red without flooding, they go extinct within a couple years. They need the flooding in order for the seeds to germinate. And then in the blue, we simulated using realistic data, weather data, we simulated realistic flooding patterns that were variable from year to year. And you see that the population varies over time. So this is just to show you how we can incorporate different key factors, chemical and non-chemical, influencing different aspects of the species biology and ecology to look at the population dynamics over, in this case, temporal scales that would be hard to do empirically, which is another benefit of the models. So in this case, the flooding was essential for species persistence. The herbicide effect was relatively minor, but we did some simulations where we could shift the competitive balance between this species and others, and that influenced the outcome at the population level. So how can the mechanistic models help? And I'm concerned that you're not holding up your end of the deal, because so far I'm on what, slide 5, and nobody stopped to interrupt me. And I'm getting a little concerned about this. So.
Excuse me, Valerie, you're just being too clear.
Liar. All right.
All right.
Yes.
When you go back, I guess I got confused.
All right.
Are those on the right hand theoretical output from the model, or
yes.
is it empirical data?
Yes. This is showing theoretical output, though we have, and I want to touch on that too, because I think some of our validation challenges at this kind of scale are somewhat different than at, say, the whole cell scale, just because it's difficult to validate some of these things, like, say, population size over a 15-year period. It's rare that you may have those kinds of data, though you can validate different submodels, but these data here are predictions from the model, though this model was at least partly validated with actual field data. So I'm not showing that here. Okay, very good. Thank you for your question. Okay, so how can the models help us do this kind of work in general? More so than doing our laboratory experiments, we can look at effects of stressors at more realistic, both temporal scales and also spatial scales, and also variability in exposure. In our laboratory experiments, we kind of expose our organisms to the chemical stressor over a constant time frame. And in the field, we know that exposure is varying both spatially and temporally. Relevant scales. We can also extrapolate better from one species to the next. So we tend to do our laboratory experiments on that handful of species that we know and love, that are easy to grow and culture in the lab, but they're not necessarily the species we care about. And we might do tests on a handful, and we expect that our results for those species allow us to protect all those other species out there that we also care about and want to protect. And the models can help us to extrapolate from one to the other. For example, you could use your toxicity test data from your standard species, but incorporate that into an ecological model of a different related species that you are trying to simulate. We can also include behavior. So species sometimes avoid stressful conditions, and that can make a big difference on impacts at the population level. Dispersal and other life history features that we tend to completely ignore certainly in our traditional toxicity tests. These can all be included in the models. And as I hope I'll show you in a couple minutes with some examples, instead of just making implicit assumptions that, well, we measure toxicity and this tells us something about what we care about, because after all, survival, growth, and reproduction, which is what we measure in our individual organism tests, they are fitness-related traits, right? So they must link to impacts on populations. And yes, they do, but not in necessarily a straightforward or linear manner. And that's where the models can make this more quantitative and more mechanistic. The models also allow us to explore different management or risk scenarios. So once you have your model up and running, you can ask questions like, well, what if we got more rainfall over time? Or what if the temperature increased by 2 degrees? What would that do to the outcome? Or in our case, what if we managed exposure to the chemicals or managed the environment in a different way? What would be the implications for your populations? And you can do that pretty easily in a computer where, if you want to run simulations for 10 or 15 years, you're not really going to be able to do that empirically. In principle, one of the arguments for the models is that they can reduce animal testing, and in our field, particularly with regard to vertebrates, people are really getting increasingly hesitant to kill vertebrates to test the effects of chemicals. And the idea is, well, if you could simulate impacts in the computer, then you could avoid killing all these vertebrates. Now, it's one reason I'm glad I work with invertebrates, because no one cares if we kill them. But in practice, probably because of reasons of validation, et cetera, I think we're a long way from eliminating animal tests, at least at present. So let me give you some examples, because those of you who may not work with mechanistic models, it may seem still a little bit kind of fuzzy or abstract. So I want to go through some different classes of models that we use in this context. And the first I'd like to talk about is adverse outcome pathway models. Is anybody familiar with adverse outcome pathway models? AOPs, just one or two people. Okay, so these were developed by the US EPA back about a decade ago as a conceptual framework to help us think about how chemicals might impact organisms at different levels of biological organization, from the molecular level all the way up to the population level. And so this is kind of the conceptual framework for adverse outcome pathways, or AOPs, as they're called by their friends. The idea is that you have some kind of chemical that initiates a molecular response when it impacts a target or non-target molecule in an organism, and that at some point will result in some kind of response at maybe the cellular or tissue level, physiological response, which eventually will end up in a whole organism response, either death or reduction in growth or some process at the organism level, which will then in principle result in some kind of population level response. And when this was first developed, it looks very neat. You've got this nice domino effect. You've got these nice rectangular boxes, and they're connected by arrows, and it all looks very predictive and very straightforward. And when the developers of this were asked, well, okay, what's behind those arrows? Like, where are the models? Where's the math? Like, what? There wasn't any. It was designed as a conceptual framework. And still, many of the AOPs that exist, and there are databases that are being populated with different AOPs for different organisms and chemicals, but still many of them today remain as qualitative descriptions of processes and how they can be linked across levels of organization. With that said, there are people working on making quantitative AOPs, so this is an area probably in rapid development. Not the least because, as I mentioned, we can now generate huge quantities of omics data that are allowing some of these models to be developed. All right, so that's one class of models, and these, in our field at least, are really increasing in popularity. Another class of model called dynamic energy budget models, these link physiological, metabolic processes to individual organism performance. So these are basically energy budget models. Bas-Kooijman is kind of the father of dynamic energy budget theory, and he's written a very well-known book back in 2010, and this is pictorially what an what a dynamic energy budget model looks like. But basically, they describe the processes of energy acquisition and allocation in an organism. So an organism feeds, takes in energy, some of that energy is assimilated, and then it gets distributed into the organism's own growth, maintenance costs, or its reproduction. That's kind of the basics. The DEB theory follows a general set of rules that obey the laws of conservation of mass and energy. So it's often said that DEB models are based on first principles, which makes them very satisfying in a way and very generalizable across all organism groups. So in that sense, you use the same model structure for any different taxonomic group that you're working with, which makes them nice to use. They are an acquired trait, and some of the terms in the DEB models don't relate obviously to things you can measure in a critter, and that makes them somewhat challenging, but they do have a number of advantages. Nika?
Just a quick comment.
Yes.
We actually have a webinar on dynamic energy budget
Oh,
theory
excellent.
on our website.
Oh, excellent.
Made by the courtesy of Roger Nisbet.
Oh, very
Who
good.
is, if Buzz is the father, then I guess Roger is something like an uncle to the theory.
At least.
So I would suggest that if you're interested more in that theory and its applications, he I think has three examples of how it's been applied for different systems and for different questions. I would suggest to go to our website and find the webinar.
Yeah.
So you said puts are, or that there are aspects of it that are not definable, but I'm looking at this and food, feces, maintenance, maturity, reproduction, growth, those are all quite measurable.
Yes, they are, but maybe I spoke wrong. I would say the parameters in the model, and I'm looking at Nika because she knows a lot more about DEB models than I do.
Yeah.
I mean,
Well, the thing is also that some of these things are abstract. For instance, the reserves are
Yeah.
an abstract term.
Yeah.
You cannot directly measure them because it's not necessarily things like we think of reserve as lipids. It's not really just the lipids. It's all different kinds of molecules that add the structure and the energy to the growth of the organism, to the metabolism. So there is essentially a layer of processes and parameters that are abstract, and then you need a set of parameters that translate them to measurable quantities. So things like you would normally measure any morphometry, anything that you could measure in an animal gets translated into the DEB world essentially. So it's all transferable and you can translate it, but some of the things are, especially the initial phases, are pretty abstract to capture.
yeah. I mean, they do run. There is kind of a DEB community, and they run annual courses, online courses and on-site courses to get people, and some of my postdocs have taken them to get up to speed. There is a database, an Add My Pet database, which now has hundreds of species that have models parameterized, some very well, some less well. So that's kind of helpful too, but they take getting used to. They definitely take getting used to. But from a physiological perspective, if you're working on effects of stressors that might influence, an organism's feeding rate or its growth or its reproduction, these are relevant models to look at. So, all right. And they can apply to pretty much any stressor that would affect a metabolic process. You can look at with a DEB model. They've been used quite a bit in ecotox. So they do have some advantages. All right, another class of models that link individuals to populations are, of course, matrix population model. And if Bas Koyman is the father of DEB theory, probably Hal Caswell gets that nomination for matrix population models, and he's written the Bible of matrix population models. But basically, these, please come back. So in matrix modeling, a population is divided into age classes, stage classes, size classes, where every individual in that class is assumed to be identical. Okay, and then the data that's used as input to the matrix models is information on survival, fertility, and sometimes growth. But basically, you need age or stage specific fertility and survival, which is typical data that people collect in the field if you're doing population censusing. This kind of shows you very simply what they look at. You have this diagram here, and I apologize for the pointer, with the circles connected by loops. This is a typical life cycle graph that's used to depict conceptually matrix models, and you basically use matrix algebra to multiply the abundance of individuals or classes at time 1 with your fertilities. So your fertilities are along the top row, and then your survivorships are along the subdiagonal. You use matrix algebra to get your population size at the next time step. This example is pretty simple. You can make these really complex if you want. You can add spatial structure. And have metapopulations, so separate populations that interact with each other via dispersal. You can add stochasticity and transient dynamics. The math gets pretty interesting if you want to add those complexities, and you do have to be pretty good at math, but they are probably the most widely class of population model used today, and there's a vast literature on these. So the last category of models that I want to touch on, and Jonathan referred to these as agent-based models, that's pretty much a synonym for individual-based models. And again, this is an example on the left here of a picture. You may not be able to read all the text, but often IBMs are depicted in this way, which is, it's kind of a rule-based kind of thing, where every individual in the population is simulated explicitly. So all individuals are unique. And the individuals in the model go around, depending how you model it, and do what individuals do in a population. They can feed, they can reproduce, they might interact with each other, they might get eaten by a predator, they might die of other reasons, and you have rules that you put into the model for how your individuals interact. And then the idea is that the population dynamics emerge from those individual level interactions and interactions of the organisms with their environment as well as with each other. They're really flexible, whereas matrix population models, they're kind of stiff. You can only change so many things in them. IBMs are infinitely flexible, and they can be made really realistic. It's a lot easier to add spatial and temporal variability into them. The downside is that because they can be made very complex, with increasing complexity, they tend to become more of a black box. And in the work that we do, where we're dealing with regulators, and we're hoping that the models will be used by decision makers who aren't necessarily modelers, the more complicated the model, the less they feel comfortable using it to make a decision. So a lot of the work that we've been involved with has been trying to make these kinds of complicated models more transparent, more systematic, more understandable to non-modelers to increase confidence in them. Any questions? Comments? Random thoughts that are passing through your head?
So how well do these models actually work in the wild? Like if you develop your model and then you see these changes that you incorporated into your model actually happening in a wild situation, do they predict populations? I
Sometimes,
mean, I was thinking of your matrix population models, which doesn't include individual variability in them.
Correct.
And we know that there's huge variability in how good of a mom this particular animal is
Right.
as compared to the one living next door, or age of first reproduction, and
Right.
all of these other variables.
It varies, right? Some of them fit really well, and there are examples in the literature where people have validated the models to a great degree, others where there's very little. And it depends on, well, you know, that it raises some good questions, because how good a prediction do you need? And that depends on why you're doing the modeling to begin with. What time scale do you need to validate your model? Is it just for, you know, that you want to see if next year is close to this year, or are we talking about doing simulations for 100 years into the future? Well, we can't really validate those, right? So it's complicated to answer that question, I think. And I think it also underlines that we don't just run the models and, you know, go home and say, there, we're done. You know, you kind of have to do an iterative, and I guess Nika will talk about this tomorrow a little bit with regard to the modeling cycle, that ideally, and I mean, John, your systems are ideal for this too, that you don't, it's not either model or do the experimental work. You kind of do them iteratively, and one feeds into the other and helps improve. The model helps inform what data you have to collect, and then the data that you collect helps to improve the model. And ideally you do them together. So, yeah.
What kind of data do you need to feed the model? Like, there is a strict that you need always to start running that.
For this kind of model, it completely depends on your system and the question, and it depends on your critter that you're modeling and what kinds of processes really matter. So the plant model I showed you in the beginning was the Boltonia species, that they're plants, so they're not moving, they're not really interacting, so we could ignore those things, but we really had to incorporate flooding, because for that species that was really important, and so we needed to get rainfall data and flood data from the USGS. Other species and questions, you could ignore that sort of thing. So it really depends on your question. So this one here, which you can't see, this is for a small collembolan that's running around, or population of collembolans running around on the surface of a soil. And these things are moving around, they're deciding, so you start the model, and depending on what their energy level is, either they decide to feed or they don't feed. Either they survive, and this is a stochastic thing you put in the model, they survive or not. If they survive, they get older. If they're, depending on how old they are, if they become, at some point they're going to become mature, and then they'll lay eggs, and how many eggs they lay depends on the density in the population. So you'd put in a density dependent function. And then depending on when they reproduce last, they'll either decide to reproduce again, yes or no, and then the eggs will hatch, and then the cycle repeats itself. But that's very much informed by the biology of this particular organism that we wanted to model.
But for example, for the foraging behavior,
Mhm.
that baseline energy or that threshold is determined experimentally, so you already have the data of knowing, like.
Ideally, yes. Ideally, that would be how you'd parameterize the model. So you would know, what determines when the organism feeds again. And you would hopefully have some kind of physiological data, so you could put in a function or a decision for the organism. So, I mean, one of the criticisms of some of these models, particularly the more complicated ones, is that the data requirements are high, which is true. But what you're doing, you know, if you don't have the data and you're not modeling, well, that's no better either. You're kind of just guessing. So, yeah.
I have just a quick comment on that, is that if you don't really have empirical data, observations, or your own, you know, expert knowledge, you can always use theory.
That's true,
To kind
yeah.
of fill in the gaps. So the ideal, the foraging theory, ideal distribution, dynamic
Yeah.
energy budget, metabolic ecology. There are
That's
a lot of theories out there that have been developed by observation, maybe other organisms or species, and that you can kind of then just plug in into the data gaps.
Yeah. Thank you. Good point. Yeah.
Yeah, but in the same direction, I was going to ask, because you mentioned that you can even model species that are not usually approached experimentally.
Right.
So how would you get those parameters? That's probably...
Well, yes, the same way. I mean, so, and what I mean is, so from our point of view, we're testing the toxicity, we're looking at the effects of chemicals on species. We have toxicity data for for a couple of species of fish. And we care maybe about species of fish that are endangered. You can use the tox data for the fish you've tested, put that into a model where you've included the life history, the ecology, the habitat of your endangered species, and in that way it helps you extrapolate from your tested species to other species. With care, it must be said.
Thank you.
So, and I'll come back to that, because a lot of what we're doing now is working with endangered species, and there you have almost no data, which is really, really challenging. All right, so limitations and obstacles. The models can't tell you what to protect, right? They can't tell us what we should care about, what impacts are important. You have to make those decisions otherwise. And I don't want to give you the impression that these things are easy to develop. They take time to develop. A good, thorough, well-tested model takes a lot of effort. There is this perception among non-modelers that the models increase the uncertainties. And the reason there's that perception is because the models help you to make those uncertainties explicit. It doesn't increase them, it just means now you can see what they are. And that, I've heard that so many times, and it's really frustrating. So not true, not true. Yeah.
Uncertainties in what? Like outcomes or?
Outcomes, I would say. Yeah, so for example, if you're doing standard, toxicity testing and you calculate some lethal concentration that kills 50% of your species, it looks really certain, right? Whereas if you ran that through a population model and you would get some kind of probabilistic estimate that depended on different things that were in your model, it looks like your model is less certain. But in fact, all that stuff that you're ignoring when you just look at the toxicity is actually incorporated in your model, so you can see it. And you can see how important all those other things are, which you typically ignore. There are, with the more complicated models, there can be a lack of transparency, and that, again, can lead to mistrust by people who might be using the models to make decisions, and that's something that we really need to work on. And validation, the V word, we can spend a lot of time talking about the V word and what is sufficient validation and what it means to actually validate a model. And I don't have time to go into that today, but it's something that it needs care. In our field, there's been a lot of progress in using mechanistic effect models for ecological risk assessment. When we started, or when I came into the picture in this back in the early 2000s, this is really dying here. Those of us doing this felt like a lonely voice in the desert. And thanks to some major collaborative initiatives, a number of workshops of kind of like this kind of workshop, these are just pictures from different workshops, and there are publications and books published as outcomes of those workshops. We had a major this was when I was in Denmark, and we had a Marie Curie initial training network, which allowed us to train something like 20 plus PhD students in mechanistic effect modeling. That really made the difference, because all of a sudden we had a critical mass, and a critical mass of young scientists who were trained in this area, who now, and that went to 2014, now many, if not all these people, are now out working in academia, working in industry, in the consulting firms, in the government agencies. It's really changed the way that these models are being perceived in our field. And finally, as of probably a couple years ago, 2015, one of the first mechanistic models was actually incorporated in a risk assessment. It's been a long haul. And a lot of challenges along the way, and it's required us building a community of people with a common understanding. And I think there might be some important take-home messages for this group. The last effort, the most recent effort, was a Nimbus initiative. How many are familiar with Nimbus? Okay, a few more people there. So this is the National Institute for Mathematical and Biological Synthesis. And I'll come back to that in a minute. I have one slide here. So what we've realized through all of this work over the years is when we try to do these models, it really underlines how little we know about most species. And it's kind of, it's a little bit depressing. But I think that doing the modeling has also helped us to think systematically about where we need to be strategic and where we need to fill the data gaps. We're not going to be able to model everything everywhere all the time. So how do we prioritize? We've got the tools. The tools are there. These categories of models that I showed you, there's extensive literature with case studies showing the existence of different models. I don't think we need new tools to link organisms to populations, but we do lack an kind of a consistent framework. And in our field, because we're not just doing it for the sake of the science, but doing the modeling to incorporate into decision making, we also need some kind of framework to help the models get incorporated into the regulatory process. And that is a non-trivial task, which I'd be happy to talk about at length, but not right now. And this is what we're doing right now. I think this emphasis on the high throughput tools, because of these amazing advances in the technology, is offering a lot of opportunity to generate data, but those data are even further removed from the populations that we want to protect. And so there are increased challenges there for making those linkages in a way that are robust and mechanistic, and that we can depend on. And in some cases, I think we've been guilty of, the attraction to the shiny new toy. Oh, we can measure it. Let's do that. It must mean something. And often that's not the case. Okay, so I wanted to say something about the Nimbus work that we did when I moved back to the US in around 2011. I wanted to sort of help bring what we'd done in Europe to the US regulatory situation, which is very different from the European one. So Nimbus does fund, and this is maybe something to keep in mind. I think they're still funding these sorts of things, though maybe sort of kind of. So at that time, they funded one-off workshops where you could bring about 40 people together for the course of around 4 days to talk about issues at the interface of biology and mathematics. And this was a great session. We brought together a lot of people from the private sector, from industry, from academia, working on various mechanistic models, but also people involved in the risk assessment and decision making to get together and see how we might bring things forward. That led to further work because Nimbus also funds working groups. Come back here, come on. And so we use the output of the workshop to develop proposals for 2 working groups, and for various practical reasons, because you can only have 12 people in a working group. And when you're trying to cover molecules to ecosystems, that doesn't really leave you a lot of expertise. at any level. So we decided to divide up into two parallel working groups. So we had a molecules to organisms group and an organisms to ecosystems group. And it was unlike this exercise, this was the same 12 people that met over the course of 2 years for 4 meetings in total. For like a week each time or 5 days each time. And we tried to develop these working groups in parallel with common members between the two groups so that we could coordinate. And the idea was to develop, here's kind of the conceptual framework that we used, was to have our 2 working groups work in parallel, and ours, the one that I led, was tasked with developing some case studies to look at how we could link organism responses to chemicals up to ecosystem service delivery through populations and communities. And then the other group, the molecules to organisms, was going to develop some models, basically adverse outcome pathway models that would link molecules to organisms. And our grand plan was that we were going to meet in the middle at the organism level. I think this thing is ready for lunch. Here we go. And we were going to use dynamic energy budget theory to make those linkages. And that sounded great. And that was what we were going to do. And we were going to use the same case studies, same species, same chemicals, so that we could really make these linkages tight. Well, let's just say it didn't quite work out as we planned. But a lot of good work came out of it, and I think we did make some progress. What we found out is that instead of nice, neat boxes connected by arrows, the situation is a lot messier. And so it's kind of more like this, and really, really a lot of challenges, and not as easy to make. In fact, we thought dynamic energy budgets were going to be a great way to meet in the middle, and it turned out from the adverse outcome pathway side, the molecules to organisms, that wasn't quite so easy, and I've kind of lost touch with where they are. I don't know if you...
Well, they have developed some new, they have modified or added something to the theory, developed dynamic energy budget models that are integrating some of the adverse outcome pathway work and terminology and pathways. It doesn't necessarily go to the macromolecular interaction, but at that level, I think the AOPs are quite abstract. So you would have things like key events, and then chemical would initiate a key event, and that was translated into dynamic energy budgets.
Yeah.
And they've had a couple of interesting publications last year and this year.
Yeah.
But
So-
there was also, I think, the timeline mismatch that we just could
Right.
not
Yeah.
meet and actually do that because they were doing things slower or in a different pace than us.
Yeah, yeah, it was challenging. Yeah, was that?
So I don't know if this is just a heuristic or it actually represents what you were doing, but if it does the latter, why are you skipping steps in a lot of these arrows?
Well, give me some artistic license.
That's what I was thinking,
That's
yeah.
just for demonstration purposes. When we were, so, and this is sort of a private joke between Nick and I, because when I was first being very critical of the adverse outcome pathways, for saying, well, all this is boxes connected by arrows. And Nika said, Valerie, this looks an awful lot like boxes connected by arrows. And I thought, you're right. And so I tried to draw it more realistically. So that was just my humor. So let me just briefly show you what we did. So in our attempt to link organisms to ecosystem services through populations, one of the first things we did was to think sort of conceptually, how do we do this? And so if you think about, we have organisms, we have different levels from suborganisms, organism populations, and then food webs. You might have data, at least ideally, you have data at the different levels, and then the idea with the models is that they help you connect between the levels. So your dead models connect your metabolism to your organism biology, then you have population models that connect your organisms to populations, food web models, etc. You have an environment in which your organisms exist, and then for our purposes, we were interested in taking this further to ecosystem services and the beneficiaries, and we don't really have to talk about that today. We did this by developing a couple of case studies, which I hope will make things a little more concrete. So in the one case study, we were concerning our ecosystem service, or what we wanted to protect, were catchable fish in a mountain stream, and people were interested in catching fish, but also interested in having fish there. So we were looking at, the case study was a Colorado mountain stream, and the particular trout species, which is the greenback cutthroat trout, is the state fish of Colorado. And people care about the state fish of Colorado, so they want it to be there. And we were looking at the effect of EE2, which is the estrogen in birth control pills that gets into wastewater streams and gets out into the environment. And there are concerns that this could influence the population dynamics of these fish. We used an individual-based model, which is called InStream, which is a fairly detailed and complicated individual-based model that has one that's been well-validated, tested, used in real environmental decision-making for many years. Steve Railsback developed this model. The second case study was a lake or reservoir in the Midwest where our ecosystem services were clear water for swimming. People like swimming in clear water. You don't want to swim in that green gunk, right? And catchable fish, again, our stressor in this case was an insecticide that was being sprayed on the surrounding agricultural area. And here we used an ecosystem model called Aquatox, which was developed by the US EPA. And again, we use these models because they were established, published. We didn't have to develop the models as part of this exercise where we were meeting 4 times in 2 years, and it was beyond the scope. Just to give you a sense of the data, so for the first one, the InStream, we had some laboratory data and the red dots are from real data showing that male trout exposed to EE2 had lower fertilization success. So those were the tox data we had. We fed that into InStream, which incorporated fish behavior, and the 4 things that fish do in the model. They select their habitat, which is based on all types of habitat features, depth, flow. They grow, they survive or not, and they spawn. Those are the main behaviors of the fish in the model. And also InStream incorporates very realistic spatio-temporal variability in the habitat, so you have an actual river reach with all of its features. And food level and predation was also simulated. So we have our tox data from the lab, we incorporated it into the individual-based model, and we could look at how the effect of EE2 on 2 species of trout. So there is the greenback cutthroat trout, or GCT. This was the state fish that people cared about. It's endangered, so we're worried about it. And then BT, or brown trout, which people like to fish, it's introduced, but it's also considered invasive because it outcompetes the other trout species. So here what we look at is how increased loading of the estrogen influences the abundance of the greenbacks, which are in circles. And you can see there's only like 20 to 30 of them in the population, and the brown trout, which there are up to 300 in our realistic scenarios. And basically, so our brown trouts are much more abundant than the greenbacks. That didn't come as a surprise, but what we saw for various reasons due to the ecology and exposure, the greenbacks were more sensitive to the chemical, so they declined more rapidly as the chemical exposure went up. And we found, to make a very long story short, that in order to protect the greenbacks, the endangered one, you really had to manage the other species, whether or not you had the chemical present or not. And so it gave us a much more nuanced picture of how we could understand the population dynamics of these species in the field, and how with different simulations, and you could remove different amounts of your brown trouts and see the impact on the other population, which was a lot easier to do in the computer, frankly, than going out in the field and trying to do it. Yeah.
How do you explain the much greater variance in the greenback versus the brown trout in abundance? What factors in the model would be responsible for that?
Do you remember?
Well, I think these are also stochastic simulations. So
Right.
there were some extinctions in some of the simulation runs.
That's
due to
right.
Stochasticity too. For instance, when the population became too small, there were just random events that led to that.
Yeah.
And I think overall you can see that the cutthroat has much smaller sizes, population sizes versus the brown trout. So you have much more of that demographic and stochasticity influencing the extinction.
And they did go extinct. None of the greenbacks lived longer, or the populations didn't persist longer than 22 years, which was the length of our simulations, if the browns were present. If we remove the browns, they could exist for indefinitely, even if there was chemical present. So I think some of these is because we probably had some zeros in there, which were, yeah, thank you for that. Yeah.
I'm just curious whether there's any way in these models where you account for if a population is actually adapting genetically during the course of the simulation.
There are individual-based models that can incorporate adaptation. This one doesn't, but you can, which is part of the flexibility. And you can incorporate learning and all sorts of, I mean, these kind of individual-based models are also used in economics and other fields to simulate human behavior and things, so you can incorporate all sorts of stuff in them. So, all right, so this is an example from the other model, the Aquatox, where we were simulating multiple species, and this is just to really show you kind of what the model can give a sense of what the model can do. So we have somewhat realistic pesticide inputs over time for a period of about a decade or so, where in different years you get different size peaks of exposure that is going to have different impacts on the populations. And then we simulated, because Aquatox simulates a bunch of different fish species, invertebrates, phytoplankton. It's a fairly complicated ecosystem model. This is just to show you some of the 3 sport fish that we were interested in simulating, and the different colors represent our management scenarios. That's what MS is for. So the baseline is where we have inputs of the pesticide as simulated here, and then we reduce them by different factors, and then the green lines are when we had no pesticide at all, so a complete banning of the thing. And I mean, you could see that the 3 species differed very dramatically in their response in the same model, the same simulation, but because of both their sensitivity to the chemical, but really through the food web. So depending on what they were eating, if their food item was really sensitive to the pesticide and got depleted, then that would impact their population dynamics as well. So it led to a much more, again, complex, nuanced picture of the system. All right, very, very quickly, some of the work that we're doing now with the postdocs in my group. Maxime Vaugeois has just submitted a paper, and what he's looking at, and I wanted to focus on the questions that we're asking to give you a sense, if you haven't been working with modeling, what types of questions can the models help address. So his questions, and he's working with a couple of different species of fish, and he's asking what are the population level impacts of stressors, and this is general stressors because he's using DEB models, so he's just impacting physiological metabolic processes, so it could be anything. So what are the population level impacts of stressors that affect an individual's metabolism? And depending on whether the stressor is affecting growth versus reproduction versus maintenance or assimilation, the response at the population level is very different. And this is something that also has been seen before, but he's also trying to add an extra layer of ecology on top of this and asking whether the impacts differ if you've got a top-down controlled water body, so in other words, one that is controlled by predation, or a different system where the fish dynamics is controlled by food level. And in the latter, you have compensation. So if you kill off some of the fish by exposure to a stressor, well, there's more food for everybody left, so the impact on the population dynamics is somewhat less. That's not the case for your predator-controlled system. So that's kind of showed pictorially there. So that's what he is working on. Another project by another postdoc, Chiara Accolla, and she's developed a Deb-based model, again with building on some of the Nimbus work to look at these different trout species. And so she's looking at how sublethal effects on metabolism through a Deb type model that affect growth and reproduction in 3 different trout species, and to what extent are those individual level responses, if they're similar at the individual level, do they differ from the individual to the population level because of differences in the 3 species life history and their ecology? And the answer is it matters a lot, actually, which we were kind of surprised about. So that is ongoing work. And then 2 other projects. So Pamela, raise your hand, and Adrian. So this is work that they're doing that is co-sponsored by Syngenta and co-advised by Nika. And here we're we're interested in assessing risks of pesticides to endangered species, which is a big deal in this country. And you have to, for pesticides that are registered and put on the market, you have to assess the risk to endangered species, of which there are many. So there are 816 today listed species of plants in the US and 90 species of freshwater mussels that are listed. How do you assess the risks of pesticides to all of these listed species, and never mind the birds and the mammals and all the other ones? So what we're trying to do is figure out how we can fill the demographic data gaps for listed species, because for some of these species you kind of wonder how it is they got listed, because we know almost nothing about them. And how can we fill these data gaps in order to assess risks and develop models? And so what we're trying to do is take a trait-based approach, because we're not going to develop a unique model for every single species of endangered plant or mussel or whatever other species. So can we identify particular traits or features of species that make them vulnerable to stressors, be they chemical or other stressors, that would make them more likely to be listed? And then, assuming we can do that, then could we develop some kind of generic models that would represent an entire group of species in those categories? So that, and they could tell you a lot more about what they're doing at the poster session or at a later time. So I thought I'd kind of circle back and think, well, all right, this network is an attempt to begin collaborations, which I guess you're hoping will lead to, maybe at the very least, a collaborative paper among the participants, maybe an NSF proposal down the line, and you'll get some preliminary analyses or data. And I think Jonathan in his talk mentioned the importance of collaboration, but also some of the challenges of collaboration. So having done a lot of these different projects over the years, I thought maybe I'd share kind of some of the stuff I learned the hard way. When doing these kinds of projects, it's great. You often meet for really intensive periods. Everybody gets all excited and you're together, and then everyone goes home and you've got your own stuff to do, and it's really hard to keep things going. So I've found it really helpful that you identify a lead early on, and somebody that everybody agrees, okay, this person is going to be the kind of slave driver who's going to keep us moving forward and be kind of the nag of the group, and make sure that things are done and that everybody has clear deliverables. We also found that setting up those regular conference calls is really important, and it took us a while to figure that out with our Nimbus work, but it really made a difference it kept people moving forward. Also, having those several day in-person meetings are effective, but they need to be planned carefully. And one of the things we saw in Nimbus, even though it was the same group of people meeting and it was a small group, you'd meet twice a year. And I would find that, you'd meet and you'd agree on things, and then 6 months later you'd meet and you'd spend the first day re-going over all the stuff you'd already agreed upon, and it was very frustrating. So having those regular check-ins and writing stuff down, it can be really important. Deadlines, having people agree to present at conferences. There's nothing like having to get that conference presentation prepared to help get the work done. And somebody has to be the champion of the project. Otherwise, things kind of float and nobody wants to take the lead, and then things don't move forward. And I think in something like this, you know, it's a good idea to let lots of ideas blossom up at the beginning and to be really open, realizing that probably most of them are gonna stumble and not make it, but that in all those great ideas, maybe one or two will make it and move forward, and then people will coalesce on those. And that's it. I just want to thank the organizers of the RCN for inviting me, and thank my current and previous lab members for all of their fantastic work. And then there have just been all these numerous people that I've been lucky enough to collaborate with over the years to get all this stuff done. Because science is a team sport, right? So, all right, that's it. Thank you.
Thank you.